This is introduction to the unit Cell Morphology and Functions. There are 4 million living organisms in our planet and many are yet to be discovered. Among these 4 million living organisms, we have monerans, protists, fungi, plants and animals. Monerans includes bacteria, blue-green algae, mycoplasma. Protists include slime molds, protozoans. Fungi includes molds and mushrooms. Plants includes both flowering and non-flowering. Animals includes vertebrates and non-vertebrates. There are 0.5 million plant species discovered so far, and many are yet to be discovered. Similarly, 1.2 million animal species have been discovered so far. Now a question that comes to our mind: why? There is a huge diversity in these organisms. How and why they differ in shape, size and structure, and also in functioning? The answer lies is that every organism is unique and is made up of a basic unit of life known as cell. So by definition, cell is the structural and functional unit of life. Even if we see human beings, there are different types of cells in a same body. Heart cells are different from liver cells, and if we consider the functioning of these cells, heart cells and liver cells, they differ in their functional activity. Okay. Before going to the cell theory, it is important to read the history. Sir Robert Hooke, an English botanist, he observed sections of cork under microscope and noticed hollow compartments, hollow spaces. He termed them as cells. His observations are noted in a prestigious book called Micrographia. This is important. Under the microscope, he observed cork and found some hollow compartments. These compartments are termed as cells by Robert Hooke. Sir Anton van Leeuwenhoek, he observed living cells like bacteria from teeth, erythrocytes, sperms and protozoans. Sir N. Grew, he observed plant cells. Sir Robert Brown first reported nucleus in the root cells of orchids. Sir Dujardin observed semi-fluid material and named it as semi-fluid material, and this fluid was seen around the nucleus. Sir Purkinje observed or discovered a jelly-like substance and termed it as protoplasm. Origin of cell theory. Sir M.J. Sheldon found that every plant cells are similar in structure and have cell wall. Theodor Schwann observed that both plants and animal cells are similar in structure and composition, but they differ in the outer covering, such as plant cells have a thick cell wall, whereas animal cells doesn't have any cell wall. So these two scientists contributed their work and combined their observations and formulated cell theory. These two names are very important: M.J. Sheldon and Theodor Schwann. Now, cell theory postulates all the living organisms are made up of cells. That is, they are morphological and structural units of living organisms, and so life exists only in cells. Every living organism is supposed to be made up of these basic units of life called as cells. and they are the structural and morphological units of every living organisms, and life process exists only in cells. All the life forces are carried out inside cells, so cells is the basic unit of life. Cells contain hereditary material, and this hereditary material are responsible for transmission of characters. During their times, hereditary materials like DNA, chromosomes and gene was not known to them. They believed that there is some materials present inside the cell which is responsible for character transmission from one generation to another generation. They believed it was given in one of the postulates that new cells are formed from abiotic materials de novo. That is, new cells are formed by a process called abiogenesis. Later it is disapproved in the new concept of cell theory. Metabolic activities within the cells, within the living organisms, are controlled by cells. So it is the metabolic activity metabolic activities inside the cells in turns control the whole living organisms. Each cell is independent in maintaining its vitality. That is, each cell is independent in its nature. It can complete all its life processes by itself, that is, starting from nutrition, respiration and reproduction. So it is independent in its nature. Cells are nothing but a mass. This is actually the structural definition of the cell, that cells are nothing but a mass of protoplasm, usually containing a nucleus or nuclear material along with some organelles, and is bounded by a plasma membrane. It is like they have stated, I am drawing, that if this is a cell, this is a mass of protoplasm, and it contains nucleus with nuclear material and some organelles. These are the organelles, and bounded by a plasma membrane or cell membrane. Okay. Now, objections to cell theory. New cells formed inside old cells was disapproved by Hugo von Moll and K. Nageli. They disapproved the concept that new cells are formed inside old cells. Schwann stated that cell could be spontaneously generated by a process analogous to crystal formation. Proved wrong. Schwann statement, one of his statement was that cell is spontaneously generated by crystal formation, and that is proved wrong. So Rudolf Virchow disapproved de novo concept, that is abiogenesis, which stated in cell theory that cells are formed from abiotic materials, but Rudolf Virchow disapproved this and stated that cells are not formed from abiotic materials. Instead, cells are formed from the parent cells, that is pre-existing cells, which is called as the theory of lineage, which states omnis cellulae cellula, supported by Louis Pasteur, that life originates from pre-existing lives, biogenesis. Exceptions to cell theory. In the structural definition given in the cell theory, it is stated that cell is a mass of protoplasm covered by plasma membrane having nucleus containing the nuclear material. But you see, viruses are an exception to cell theory because viruses lack protoplasm, but they are made up of proteins and one of the nucleic acids, that is DNA or RNA. Bacteria and some algae lack well-developed nucleus. Bacteria and some algae, their nuclear materials are dispersed in the cytoplasm rather than confined in a nucleus. So this is an exception to cell theory, because in cell theory it was stated that cell nucleus is well developed in any cell, but bacteria and some algae, they don't have developed nucleus. Cell principle or cell doctrine. It is a better version of modern version of cell theory. Now what it in brief, we can say that cell principle or cell doctrine is stating that cell is the structural unit of life, cell is the functional unit of life, and new cells are formed by the growth and division of the pre-existing cells. That is, new cells are formed from the parent cells. Three important regions of cell: plasma membrane, cytoplasm, and nucleus. So let's draw. If this is a cell, this is a plasma membrane, this is the nucleus, total is the cell. So plasma membrane is this outermost part or region of any cell. It is outermost, thin, transparent, electron microscopic, that is, it is not visible under normal microscope and can only be visible under electron microscope. It is self-regenerative, semi-permeable, elastic, that is, it permits only special type or some type of substance in to and fro from the cell. So it is semi-permeable and elastic. So functions of the plasma membrane: exchange of materials in and out of the cell for maintaining its metabolic activity inside the cell. In plant cells, outside to plasma membrane, there is a rigid layer, and that rigid layer, which is lacking in animal cell but present in plant cell, is known as the cell wall. This green color is the cell wall. Okay. Cytoplasm. If this is the cell plasma membrane, this is the nucleus. So between nucleus and plasma membrane, this colloidal, amorphous and homogeneous substance, this yellow substance present between the plasma and the nucleus, not actually the yellow substance, I am just drawing it with a yellow color. So this, to make you understand, this thing is the cytoplasm. This is colloidal in nature, amorphous and homogeneous. Why it is homogeneous? Because its constituents is uniformly distributed around the cell, inside the cell. Function of this cytoplasm: it is the area or the site where biosynthesis takes place. That is, biosynthesis means protein synthesis or protein formation takes place, and all the catabolic pathways, that is cell respiration. Many cytoplasmic structures are suspended in the cytoplasm, and many cytoplasmic structures means it can be either cell organelles or cell inclusions. Cell organelles are living and cell inclusions are non-living. These are together, these are actually cytoplasmic or structures, that is cell organelles and cell inclusion. Cell organelles, example, endoplasmic reticulum, ribosomes, Golgi body, mitochondria, plastids, lysosomes, microbodies. Whereas plastids is not found in animals, it is present only in plants. And cell inclusions, which is non-living, is example is vacuole. So these two are cytoplasmic structures which is present inside the cytoplasm. Now the nucleus. this part, I'm just drawing this red part to make you understand. This is the nucleus. This is a membrane bound structure, means it is also covered by a membrane known as the nuclear membrane, and inside the nucleus chromosomes or genetic materials are present. It is the brain of the cell, and function, it gives the direction for all the cellular activities. That is, all the cellular processes are governed by the nucleus. The chromosomes containing DNA forms the genes which acts as unit of hereditary. So based on the type of the nucleus present in the cells, cells are categorized into two types. One is prokaryotic cells, another is eukaryotic cells. Pro means old and eu means new. All the unicellular organisms like bacteria, they are prokaryotic cells, and human beings, we are made up of eukaryotic cells, that is new karyotic cells. The karyon means nucleus, pro means old nucleus, and eukaryotic means new nucleus. It is actually if the nuclear membrane is present, it is called eukaryotic cells, and nuclear membrane if absent, it is known as prokaryotic cell. So the differences between prokaryotic cells and eukaryotic cells. Prokaryotic cells, nucleus. The nucleus in the prokaryotic cells is not enclosed by any membrane. Means the nucleus is mixed with the cytoplasm and the genetic materials are dispersed in the cytoplasm. But in eukaryotic cells there is a well developed nucleus and the nuclear membrane is present, and genetic materials are confined in the nucleus in the eukaryotic cells. In prokaryotic cells, cell wall if present contains muramic acid, and in eukaryotic cells, cell wall is variable, and if cell wall is present, muramic acid is not present. Photosynthetic apparatus in prokaryotic cells, thylakoids if present, free in the cytoplasm, and in eukaryotic cells when present are grouped inside the chloroplast. This terms thylakoids, chloroplast, I will explain in detail in my later videos. This is just a difference you should remember. I'll talk and discuss every points which is not known to you in my later videos. So this unit will give you a full concept of the cell structure, theory, cell division, everything. Okay, so in prokaryotic cells, thylakoids are free in the cytoplasm. Thylakoids are the units of photosynthesis, but in eukaryotic cells, thylakoids are grouped inside the chloroplast. In prokaryotic cells, the organelles, cell organelles, as I discussed in my previous slides, like endoplasmic reticulum, Golgi bodies, etc., these are not bound organelles. No membrane is present around the organelles in prokaryotic cells, whereas in eukaryotic cells, membrane bound organelles are found. In prokaryotic cells, ribosomes are found free in the cytoplasm and it is of 70S type, whereas in eukaryotic cells, ribosomes are, if present, free in cytoplasm or associated with endoplasmic reticulum. Both 80S and 70S type of ribosome is found in eukaryotic cells. Centrioles are absent in prokaryotic cells. Centrioles are present in eukaryotic cells, especially in animal cell. Centrioles, what is centrioles, I will discuss in my later videos. In prokaryotic cells, no streaming movement of the cytoplasm is found, which is termed as cyclosis, and in eukaryotic cells, cyclosis is present, that is, cytoplasm shows streaming movement. Plasmids and pili. In prokaryotic cells, plasmids are present, whereas in eukaryotic cells, plasmids are absent. Plasmids are extracellular DNA present in bacteria, and I will discuss. Flagella in prokaryotic cells is made up of single strand. Eukaryotic cells 11 strand if present. In prokaryotic cells, size ranges from 1 to 10 in 1 to 10 micrometer, and in eukaryotic cells it is 10 to 100 micrometer. Spindle formation. Mitotic spindle is not formed during cell division in prokaryotic cells, and in cell division mitotic spindles are formed in eukaryotic cells.